Guys, Mr. Hyatt here. This is part 2 of the chapter 14 lecture. We're again in this video picking up the vocab. Phenotype is the physical appearance of the organism. The genotype is going to be the genes. So if I ask for a genotype, I'm asking for letters. If I am asking for a phenotype, I'm asking for words. Examples: phenotype on the left there, you see 3 purples and 1 white. Genotypes, we see 2 different, some homozygous and some heterozygous that still lead to purple, and then we see the homozygous recessive. That's our double recessive. So again, hopefully that's review. Homozygous means they're the same. Homo always means same. Hetero always means different. So the 2 alleles are different. In a heterozygote, there are 6 possible Mendelian crosses. These are things that you should probably commit to memory. I don't know that I would like flashcard memorize these, but it definitely helps. I was someone who, as a student, I said to myself, well, I know how to do Punnett squares, so I'm not going to mess with memorizing all this. I'm just going to go the long way and do the Punnett square every time and make sure that I'm right because I have that basic fundamental skill and I know how to do that. That worked for me, but it definitely made things more difficult, and there were times that some of my, I would run short on time on tests because I would have to go the long way. So I really want to advise you, if you don't memorize these patterns with me, do it when you get to college. When you take a biology class in college, it is beneficial. It's sometimes annoying. I'm against memorization as much as possible, but it really does help. That when you see you got 2 homozygotes that are opposite, you know you don't have to do the Punnett square because they're all going to be big T, little t. Most of us are probably smart enough to figure that one out. And we were probably smart enough to say, okay, look, big T has to be a big T, has to be a small t. Okay, so they're all going to be big T, little t. There's a big T in that genotype, so that means the phenotype is going to be all dominant. This one we probably recognize because we've done it so many times. This one is super easy. This one is super easy. These 2 are the ones that sometimes can be somewhat tricky. So test cross is a big vocab term that tons of people missed last year. So a test cross is going to be heterozygote if you think it's a heterozygote. So you're trying to test to determine what is that second allele of your suspected heterozygote. Well, the best way to figure that out is to cross it with a homozygous recessive, because you know this parent is going to give you a little t. So if all of your offspring have the dominant phenotype, then you know this second allele has to be a big T. Because if there's a little t here, you're going to have a 50-50 ratio. You're going to have, sometimes you're going to get big T, little t, sometimes we're going to get little t, little t. So don't forget that term. Lots of people got to the test last year and were like, what's a test cross? I don't know what that is. Well, that's what I'm asking you. That's the point of the question. So yeah, there's kind of the Punnett square of that. If that didn't make sense, go back, re-listen to that description, take a look at the picture. I think it's pretty straightforward. A dihybrid cross, you've done these before. These are bigger Punnett squares. These are the 16 boxes where you're going to do a Punnett square with 2 genetic traits. We're going to spend some time with these this week and next. You're going to need 4 letters to code for the cross. Each gamete has to get 1 letter for each trait. So notice we don't have TT and RR here. We have TR, TR. We'll get lots of practice with dihybrid crosses. We'll even dabble in some larger Punnett squares. We'll talk about those later. So number and kinds of gametes, that's huge. How many possible gametes are there? How many, what is the ratio? Are we 50-50 with big T, little t's, or are we 75-25? Those types of things are really, really big. So the formula 2 to the n can be used where n is the number of heterozygous traits. So if we've got 2 heterozygous traits, 2 to the second is 4 different possible gametes. If we had 3 here, n would be 3, like TT, RR, UU. Then n would be 3, so 2 to the third, 2 times 2 is 4, times 2 is 16. Wait, 2 times 2 is 4, times 2 is 8. So there are 8 different gametes in that possibility. So then your Punnett square is getting really huge, and things are just getting ridiculous at that point. That's where you need a data analysis, Microsoft Excel, something along those types of, along those lines. So, cross is a 4x4 here with our two heterozygotes. Each parent can produce 4 gametes. If we do the cross, we end up with that. 9 tall and red, because that's dominant. 3 tall and white, dominant, recessive. 3 short red, recessive, dominant. 1 short white, recessive, recessive. So that's our 9-to-3 to 3-to-1 ratio. Okay, law of independent assortment. We know this already, right? Like, independent assortment says that the first genetic trait doesn't influence the second genetic trait. Just because I get my mom's chromosome 1 doesn't mean I'll get her chromosome 2. So that's true even in the case of a dihybrid cross. You can burrow down. You can kind of dig down a little bit. I can ask you to do a dihybrid cross, and then I can ask you questions specific to one of those. The ratio of tall to short is still 3-to-1. You count up those boxes, it's still a 3-to-1 ratio. Ratio of red to white, still 3-to-1. It's a dominant recessive relationship. The cross is really a product of the two multiplied together. So remember that when I start talking about some of those monstrosities, right? We can do 20 traits if they're all 3-to-1. 3-to-1 times 3-to-1 times 3-to-1 times 3-to-1. And eventually we'll get our 9-to-3 to 3-to-1. Okay, here's the demonstration of that. So here's our independent assortment where they're unlinked. This is false, so we don't want to mess with that. Look over here. Here's all of our different gametes. We just drop them down, take them across like we would a basic Punnett square. And then what they're trying to do is show shriveled. So they're doing seed shape and seed color. Those are their 2 traits here. And you can see if you were to add them up, you're going to get this 9-to-3 to 3-to-1. If you add up smooth to wrinkled, you're going to get 3 smooth to 1 wrinkled. If you add up yellow to green, you're going to get 3 yellow to 1 green. So our patterns hold. That's what this stuff is all about, guys, is patterns. If you're somebody that naturally sees patterns, you're going to do well with genetics. If you're not, look for those patterns. So genetic is, like it says here, so that's just an extension. It's a specific application of the rules of probability. If you haven't gotten a probability in math yet, it's essentially your likelihood of having a possible event. So you know that the coin flip is 50-50. That's how this is trying to demonstrate it. What are your chances of having each individual gamete, and what are your chances of having each individual zygote or offspring? So these monohybrid ratios are actually the probabilities. So if we get a 3-to-1 ratio, that means we've got a 75% chance of the dominant. That's our 75% probability that we're going to have dominant, and 25% probability we're going to have a recessive trait. Kind of mentioned this a few minutes ago, so I'll go through it really quickly. Let me just skip to this slide. So when we do larger crosses, sometimes it's easier to just do the multiplication than it is to try to lay out a massive Punnett square and drop them all down and take them all across and all that kind of stuff. So if we look at this, we know that if we look at just the T's, we've got 2 heterozygotes. So if we were to lay out that Punnett square, we'd end up with tall being 3 out of 4. Same thing with red, 3 out of 4, because it's the same setup, 2 heterozygotes. So we multiply 3 out of 4, 3 out of 4, we get 9 out of 16. So the probability of getting a tall red offspring is 9 out of 16. If we wanted to do short red, we would take 1/4 times 3/4, and that would give us our probability of getting a short, I can't remember what I said, short red, I don't know. But I think you get the idea. So you can use the product ratio to calculate results way easier than you can with Punnett squares. So there's an example. Work through that on your own, and we'll talk about it in class. We'll go through this. We'll do the product rule, and we'll do the Punnett square, and we'll kind of talk about what's easier, I guess. Okay, so yeah, Yeah, this is what I talked about earlier. I think I said it pretty well earlier. Dominant doesn't necessarily turn off the recessive allele. The alleles aren't right next to each other. They don't influence one another. They're just different. So one often masks the other or covers up the other. Okay, so there are some variations on Mendel. Like I said, it has to be either or for Mendel to work. So when the trait is not either or, when there are multiple possibilities, something else is going on. This is really where genetic research has been focused since the mid-1900s. We're trying to figure out, okay, we got the basics. We understand how the basics of inheritance work. We get dominant recessive. But that doesn't apply to some of these complex traits. Look at human height. There's a range from really, really short to really, really tall. There's no either or. So we're going to talk about each of these coming up. Let's just dive in and talk about incomplete dominance. Okay, so incomplete dominance is what we talked about with blending. Okay, so incomplete is where they mix together. That happens sometimes in nature. Snapdragons are kind of the classic example. If you take a red snapdragon and a white snapdragon, again, that are true bred, we know they're homozygous, dominant and recessive, we get all pink flowers. So we truly get a mix of red and white. And then in the next generation, we get 2 pinks, 1 red, 1 white. So here's our Punnett square. We're seeing independent assortment. We match the Punnett squares. So we've got blending when we've got a red and a white allele. But when we only have red alleles, we have red. When we only have white alleles, we have white. When we have both, we have pink. Again, this pink trait, the pink phenotype, doesn't change the white genotype. It's just being expressed at the same time. So they blend together. The result of that is we get no hidden recessives. So you see every trait. 3 phenotypes, 3 genotypes. Like I said, this is the second time you've seen dose effect. So like shades of colors. Things along those lines. There's carnations, same kind of thing. Codominance. Think like how the NBA is set up now. When I was a kid, most teams had 1 star, and every team's best star would go against each other, and then the best team would win at the end. Well, nowadays, every team's got 3 stars, 4 stars, 5 stars. Maybe not every team. The teams that can compete for the championship have many stars together. The idea behind that is you bring them together, and LeBron James and Kevin Love can both dominate together. They can both be really good and be themselves and be a really good team. They'll make their team better because they are both stars. Translate that to genetics. You get a red allele and a white allele. They're not mixing together, just like the NBA players don't become one dude. They don't mix together, they dominate together. So you're going to end up with, instead of red and white making pink, you end up with spots of red and spots of white. So as a result of that, again, there are no hidden recessives. There are 3 phenotypes and genotypes, but there are no shades. They're polka dots. There's a type of cow called a roan cow. This picture shows what I'm talking about with roan cows. So we've got a cow that's red, it's pure breeding, and a cow that's white, it's pure breeding. We mix them together and we end up with this color that's roan. So it's kind of like spotted. There are places where it's red and there are places where it's white. Again, they dominate together. They don't blend and mix together. So we'll call that the end of video 2. We'll pick up there with video 3.